you are. My name is Caroline Hopkins. I'm a senior reporter here at Precision Oncology News, and I'll be your moderator today. The title of today's webinar is High Dimensional Cell Morphology Augments Multiomics to Advance Translational Research. Our speaker today is Dr. Peter von der Spek. He's professor of clinical bioinformatics at Erasmus University Medical Center. And then later on during the Q&A portion of the webinar, we will also be joined by Kelly Miller, who's Deep Cells Director of Market Development. You may type in questions at any time throughout the webinar. You could do this through the Q&A panel, which you'll see appearing on the right side of the webinar presentation. And then if you look in the bottom tray of your window, you'll see a series of widgets to enhance your webinar experience. And there you will also see the survey located here. We'd love if you provide your feedback at the end. So with that, I will turn it over to Peter. Peter, thank you so much. The floor is yours.
Thank you. Thank you for giving us the opportunity to present our work from Erasmus University Medical Center. I'm going to move to the next slide. All right, so I'm going to talk today about the single-cell high-dimensional morphology analysis that we are performing in the Department of Pathology at Erasmus. And we have been having the opportunity to work for a period of 5 weeks with the very impressive REM-I machine from the company Deep Cell. And I'm going to show you first in my talk a sort of introduction on the platform. Then I'm going to outline 2 of the studies that we have been doing in detail, and then there will be space for question and answers. So what we basically see in this slide is the platform. So it comprises of a REM-I instrument, which you see on the top part. And then there's a human foundation model, what you use to sort and basically train your model to separate cells. And on the right-hand side, you see a sort of PC, which you use to analyze your results from your particular study from which you have basically been running your samples. So what you're doing with the REM-I instrument is basically imaging of cells. And basically, the machine has the capability of sorting 6 different wells with particular cells that it can recognize after you have trained the machine. And basically, you can real-time do analysis on these single cells for which you have identified the morphological differences. So it's pretty much of an end-to-end system, which comes with a data suite. So your learning model is in there, and basically you can upload your samples and basically analyze them. So the typical experiment comprises of a tube with single cells in it. The cells go through the machine, you image them, you can sort and extract them, you analyze your data, and then basically you can recover the sorted cells. And the machine is even capable of keeping your cells alive once they go through the machine. So potentially you can image cells and keep culturing them after you have sorted them and actually do cleanup steps. So these cells are ready to go for further multi-omic studies, which is very exciting. So typically you see the machine here in action. You see cells going through the machine. The cells can get sorted, and basically they are captured in 6 different wells, and the remaining cells, they stay in the waste bucket that you see on top of the queue where the cells are passing by. On the right-hand side, you see the different clusters. Each dot represents a photo of a single cell, and in each cluster, cells that have the same morphology upon interpretation by the AI model are basically grouped together in this kind of UMAPs. So as I said, you have your tube with your cells, you pipette the cells, you load them on the machine, you have an imaging area, so that's where's the bright field camera. So the cells are not at all labeled, you just photograph the cells without any dye, they can stay alive, and they go through the machine. And what you basically see is the cells, they get imaged at high resolution. Then you get your clusters of your cells that are, each color represents similar cells that have the same morphologies. And then you get representative images of all of these clusters on how the cells look like and basically what cell types they are and so on. These cells, after they are sorted, basically they go into this multi-well sorting, and you can actually see the colored different cell types, which you can use for further follow-up validation, RNA-seq, etc. So you can see downstream analysis, you can do cytology to map copy number aberrations, you can do SNP analysis, RNA-seq to identify which cells do have particular mutations. In particular, if you look at driver mutations in cancer research, you can have different lineages in which cells are differentiating. And of course, then it makes sense that you can see in which lineages do the mutations occur, and are there certain lineages that don't have the mutation, etc., etc. Of course, you can also do immunostaining, so antibody staining, etc., or further functional analysis downstream of your separation. Of course, if you look at a pool of mixed cells, you're way less sensitive than after you have separated out a clean fraction with single cells that are morphologically identical. And then basically you can start to look what underlying mechanism is potentially causing a disruption in the disease that you try to address. Okay, what do we want to see now?
The multiome.
So I'm going to switch
it's
to
the
movie.
next frontier in biological research. Yet high-dimensional analysis of cell morphology has been missing. Until now. The morpholome can finally be explored. It starts with basic single-cell preparation, but adds high-resolution bright-field imaging of every cell and sorting of unlabeled cells to get you from sample to publishable data in just hours. With the power of morphology magnified by AI, the possibilities are endless. Explore your newest ohm, the morpholome, with REM-I.
So again, you see here the very fascinating queue where you separate the cells. You see the cells coming along from the camera. They get recognized, and the AI immediately is sorting them, and basically you can capture the fractions for follow-up research. So what I'm now going to do is guide you through the areas where we think are the highest potential to apply the technology, and I'm going to outline 2 of our experiments that we have done for which we have very clear results. So of course you can apply it in cancer research, in cardiology, neurology, immune transplantation, and in pediatrics. In particular, one of the examples that I'm going to illustrate looks at PBMCs, blood cells. Well, of course, the blood is very important and easy accessible in diagnostics in a hospital. So from that perspective, we wanted to do a study where we look at blood. And basically, we did another study where we looked at the urine, where we wanted to study a particular type of urinary cell carcinoma, a cancer. And basically, in that case, you also can see very specific contributions of cell morphology, which actually can be experiments that can get incredibly speeded up by basically avoid having to do isolation of the cells, pour the cells in a paraffin block, cut slices, do stainings, then see which cells are positive. Here we basically push the cells through, we separate and count them, and basically in a day we have a full picture of everything what's going on, where in the other experiments you are basically talking about a 3-day type experiment. So in the group of Professor Arno van Leenders, they're studying urothelial cell carcinoma. So we wanted to study whether the REM-I machine can increase the throughput, but also the sensitivity of the detection of this type of cancer. So our goal is to improve cytology in general. And when we were working with the prototype of the deep cell machine, we even could not yet sort the cells. So we could bring them along the camera, photograph and count them, but we could not yet at that stage sort because that functionality was not yet in. Okay, what we typically did was take normal cells, atypical cells, or urinary carcinoma cells, push all these cells through the machine, image them, and then start processing the data from the different experiments and see whether we could see differences between the three groups that we were studying: normal cells, atypical, and urinary carcinoma cells. Now, what you then see in the last picture, in step 3, you validate your classifier, which is capable of detecting the urinary cell carcinoma. So after we had trained our model, we wanted to see, can we separate the atypical from the urinary cell carcinoma samples? So what you typically see here is images that are captured by the bright field camera and images that have similar shape and morphologies. They're grouped together, and basically the representative images, which are obtained by using the REM-I machine, helps you to look, and basically it recognizes textbook examples of cells that you expect to be present in the urine. So you see some textbook example and you see very clearly overlap. Now, what was for us very surprising, okay, the machine recognizes 12 distinct morphotypes. So 12 different clusters, which are illustrated by these different colors. And basically, if you look at the UMAP, so it's a statistical representation of how the morphologies in the different clusters look like. Basically, I'm telling the students, you see a bird here. And if you look very careful, the bird has here on the right side a beak, a yellow beak. Now, look very careful. After studying the three groups, we obtained every time the bird, and we are projecting on top of the bird the different cell shapes. So what you see in the middle is the normal cells from the normal, not diseased individual. And you see some morphotype 11 occurring very frequently. So the more red, the more of those cells you are actually counting and seeing. The atypical condition has other cell morphologies, whereas the head of the bird is very intense staining with cells that we know which are very clearly linked to urinary cell carcinoma. So this is for us very promising because the machine basically very rapidly is imaging, but it's not only imaging, it's very clearly highlighting the differences between the three conditions that we wanted to separate. This is the same bird, and if you look very careful, the bird on the right-hand side has a beak. That's the urinary cell carcinoma. So that orange cluster is clearly linked to the urinary carcinoma. But in the middle, the normal has on his part where are his legs, a green island, and that green island is basically absent in the atypical, but also in the urinary cell carcinoma. So each of the clusters has its own specific characteristics, which can be very beneficial to do very highly sensitive diagnostic separation of the different conditions. So you do not only have to see the cancer cells. If you see more normal cells, like these green ones, you're also knowing that you're looking at a more healthy condition. So what we typically see again is these representative clusters that are characteristic here for the urinary cell carcinoma. Now, in our first pilot experiment, where we just had three groups that we tried to separate, even without optimization, we were capable of separating with a prediction accuracy of 89.1% the atypical from the urinary cell carcinomas. So that is a very, very promising, sensitive way of detection, which shows the the power of the platform. So in summary, the platform works well to profile cell images obtained from urine samples. Numerous cell clusters, we call them morphotypes, they can be identified among the normal and the two pathological groups in the urine samples, so the atypical from the urinary cell carcinoma. AI-assisted high-dimensional morphology analysis can predict atypical and UCC cases with high accuracy at a single cell level. So our first pilot was pretty successful. And I'm going to switch gears. We're going to the second example. Okay, this is also a pathologist. This is Dr. Marianne Klaassen. And she works on rejection of kidney transplants. Okay, let's see if we can apply also the REM-I in the kidney transplant space. So to establish a minimally invasive method for the early detection of organ rejection in kidney, but also heart, liver, lung, other transplant patients can be studied. Our aim is to reduce the effect of invasive biopsies that can trigger rejection or make even the condition worse. So in this experiment, we basically used the REM-I platform, and we wanted to explore whether it's compatible with studying PBMC samples from our transplant patients. Now, our hospital has obtained from transplant experiments for years and years a huge biobank. So we know exactly from patients historically what was happening and when. So we, after the transplant, weekly PBMCs were collected and frozen down in the biobank. So we went back to our biobank and basically wanted to lift samples which could tell something about whether the patient was, for instance, having too much immunosuppression, which basically goes with infection or neoplasia. And if you have too little immune suppression, you get your rejection triggered. So in order to study that, we lifted samples from our biobank, which either fall in the category no rejection, T-cell mediated rejection, or antibody-mediated rejection. After imaging those cells, basically we look at the results in the Axon suite. That's basically the software that comes along to look at the results. And then we obtained a 75.8 accuracy, prediction accuracy to separate no rejection from rejection. Okay, so currently the case is once a patient comes back to our hospital with a complication after a transplantation, it can be either an infection or it can be a rejection. For us, basically it's throwing a coin, and with a 50% accuracy, we are going to guess what's going on. I'm showing here you, without optimization, a 75.8% accuracy in detecting whether we are dealing with a rejection or not. So that is an improvement of at least 25%. And you can imagine the cost of a transplant. Well, if you can recognize whether you deal with a rejection or not, is very important. But if you are dealing with an infection and you're switching off the immune system because you think it is a rejection, you can lose your patient on the infection the day after. So there's big risks involved in the decisions that you make. And we think that the REM-I platform can really be very valuable to assist in future once the platform is fully validated in those type of complex decisions. So here again, we see in this case 14 distinct clusters of cells. And basically we see again a map, and on the UMAP we can see the different cell morphologies. And by going to the next slide, we get again these kind of red areas where you have more cells. So it's a heat temperature type visualization. And what's very obvious, on the right-hand side, you see the no rejection. In the middle, the antibody-mediated rejection. And on the left, the T-cell mediated rejection. What's already obvious is that in the bottom of these, basically map, you can see there's rejection, where the no rejection is way more pointing with its morphotypes to the top part of this UMAP. Okay, these different morphotypes with these numbers, for each of the morphotypes, you have exactly the cell counts. So that is pretty informative, because if you look at the number, so you see the samples on the left in the first column, and then you see morphotype number 0, morphotype number 2. In terms of the cell counts, they are very much resembling each other. Now, when looking at these counts of these cells in the particular patient samples, you can already see that the morphotypes here on the right with 13, 3, 4, and 5 are really very different and opposite of the morphotype 0, 2, 10, 12, and 11. And in the middle, they are way more mixed. If we look at that same data, but then again, on top of these UMAPs, you can actually see each dot is a photo of a cell. So in the no rejection, you have a lot of red cluster where many photos of cells are present, where in the T-cell mediated rejection, the red cluster is almost gone. Now, in this way, we can very clearly separate out these different cell types, but also the contributions of these morphologies. So again, the prediction accuracy that we obtained in this experiment is 75.8%. So in summary, the REM-I platform works very well to profile PBMC samples from transplant patients. Numerous cell clusters can be distinguished in PBMC samples, and AI-assisted high-dimensional morphology analysis distinguishes transplant rejection cases with high accuracy at the single cell level. A specific morphotype, number 6, can be actually linked to T-cell biology and T-cell mediated rejection. We're following this up in independent validation studies right now. In future, we are also very much interested to study with the REM-I a process which focuses on hematopoietic stem cells. This process is called CHIP, clonal hematopoiesis, and basically that's linked to a 40% mortality, overall mortality. So after a hematopoietic stem cell is differentiating into different lineages of myeloid and lymphoid cells, we know methylation genes can get somatic mutations. These somatic mutations, they actually cause this 40% overall mortality. Now, this is very important in a new field which we call oncocardio. And in oncocardio, typically females who are being treated with chemotherapy are being monitored. And it turns out that obtaining mutations in these methylation genes leads to a pretty high mortality. So we would like to investigate these clonal hematopoiesis more in detail. And we're going to do that with Shyamala Sinadurai, who actually did a Marie Curie study for 4 years in Poland, where she basically figured out and has set up a huge biobank and has studied the implications of these DNA methylation mutations. We also have set up a house of AI because we realize if we are going to bring novel technology into the clinic, we will have to train also existing clinicians in using these new application areas and knowing what you can do with this kind of new technology. In the House of AI, which is set up at the Erasmus University, new data analytics tools are present to study these morphological changes as well. So the impact of this REM-I platform on healthcare costs. We are not allowed to increase our healthcare costs in general. So if we are going to do more diagnostics, automatically we have to basically show that we treat less but get better outcomes. So in order to go to more personalized approaches, more precision type medicine, you actually can apply this new technology in diagnostics and select only those patients who benefit of particular expensive cancer drugs. So like we studied the immune profile in the T-cell and antibody-mediated rejection, we can collect the same PBMCs and see whether with immunotherapy in a cancer treatment, we are modulating the immune system, yes or no. So you can really get way more quantitative data on your therapy response. So the platform has also very high potential in studying therapy response and effects. So I have to acknowledge some of my colleagues, Thierry van der Bos, who runs the lab in the pathology department, Sigrid Zwagemakers, who is in the bioinformatics team, Arno van Leenders, who is studying the urine samples, Marianne Klaassen from the transplant data, and Shamala Sinadurai, who has been thinking with us about future experiments and new projects that we aim doing in the coming year. And last but not least, I would like to thank the team of DeepCell, who have been really very helpful in actually the 5 weeks that we had them on site. We have been using every hour that was available to push samples through the machine to generate so much data. We're very, very excited with what we have seen, and we are looking forward to keep collaborating with them on the new developments that are upcoming. So with this, I would like to end my talk and open the floor for discussion. Caroline, please take over again. Thank you.
Thank you so much, Peter. As a reminder, if you want to ask a question, you'll see that in the Q&A box in the control panel. And yeah, we'll now start the Q&A portion of the webinar, and unfortunately we have a fair amount of time, so don't hold back. We're also joined back by Kelly Miller from DeepCell. So let's see what questions we have here. Couple questions on, the timing and the workflow. One, how much time is needed to generate the UMAP? And actually, this is sort of a two-part question, but related, will the UMAP update correspondingly during the cell sorting?
Yeah, so Kelly, can you answer this one?
No, please.
Or do I?
Yes, sure. Hey, up to you. Feel free to add on.
So we are very excited that it's pretty fast to make these UMAPs. So we typically take a day or a day and a half, but then you already have a very good feel about your whole experiment. So there's definitely a gain in time versus conventional technologies that we had to apply to come to certain conclusions. And the sensitivity turns out to be way better than what we could reach for some of the projects that I've shown to you, like the urinary cell carcinoma. We really, with high accuracy, can detect immediately the samples of the cancer group and filter those out for further follow-up. And what I also like on the question that I see in the dashboard, can you load the sorted cells again to the machine? Yeah, of course. And then only your pools of cells become more clean. So you can actually also rerun the cells through the machine for the second time, or basically run from the waste again the cell material and separate another cell fraction that you would like to collect. So it's a pretty flexible system.
Absolutely. I don't know if you had anything to add on that, Kelly.
No, just from a technical, specification standpoint, I'll just say that the UMAPs are generated in real time. And in terms of the updating, you can, of course, when you analyze the data, you can reproject if you would like to get, a more granular look at specific morphotypes and look for subtypes within that.
Absolutely.
And what we also like very much is that we could basically, if you look at your 3 groups that you compare with each other, you actually, with your downstream analysis, you can revisit how specific clusters change or differ between the 3 conditions. So you can look at a single sample, where is it falling, but you can also basically see what's the contribution of the specific cell types in terms of their biological function. So you can use the machine, of course, for research purpose, and that's what's the initial aim And that's why you can do the sorting and do downstream analysis. But also in a clinical environment, sometimes you just want to separate out cells that you think are suspicious, and then you spin them down on a glass slide and you throw them in a diagnostic machine from Roche, and you start doing, for instance, in a Ventana, a multi-staining with different dyes to see on the cells which surface markers are present, etc. You can do very smart combinations with other platforms as well. It's a very, very multi-usable platform.
Absolutely. I don't know, we take this question, but will the algorithm differentiate more subtle morphology variations? You know, if you're loading them in through again.
So
we are very surprised. Sorry, Kelly. Your turn.
Oh no, I was just going to say, yeah, you can collect the negative fraction and run those through again, and sort if you'd like to collect a different, different set of populations.
Absolutely.
What I very much appreciate is that the platform actually can see things what we by eye can't recognize. So if you have T cells, naive, and you have activated T cells, the platform separates them. Okay, if a pathologist looks through his microscope, he can't see the difference between an activated and a naive T cell. So how is the platform doing that? That's the advanced AI. So it figures out that, for instance, if you have your cell organelles, each photo of a cell has pixels. But the advanced AI can see that, for instance, if mitochondria are swollen, your eye can't see that in some of the cells, the mitochondria are slightly swollen. If they are swollen, it points towards stress. So that's the activated T cell. So the machine is learning and gets only more and more sensitive. The more data goes through, the smarter the thing gets. And the pathologist can't even see that with his eyes. So this is what I really appreciate, because if you start separating your activated versus your naive T cells and you start doing the staining or your downstream analysis, you clearly see the molecular differences between these cells.
Mm-hmm. Absolutely. And this is a related question to that, but did you find the cell morphology in the images, as expected according to your own, pathology expertise?
Yeah, so that's what we said. But in the urinary cell carcinoma, you see a lot of squamous cells. Those you really expect in the cancer condition like urinary cell carcinoma. You expect there UCCs popping, the squamous cells popping up. And those cells also frequently have mutations in FGFR3. So that basically parallels those particular cells. So definitely what it's separating is clearly in line with what you expect from the known pathology, but it also pops up new biology. So I can go back to one of the slides where we have been pretty intrigued, where we saw something what we never have seen before. So, okay, let me see if I can navigate through the slide deck very rapidly. Okay. This slide, and then I have to push to the audience. Okay. Here we see the antibody-mediated rejection in the middle and the T-cell-mediated rejection on the left. Okay, in the T-cell-mediated rejection, it says, morphotype number 6. Well, we are going to disclose something very interesting here. We found Mickey Mouse cells. We named them Mickey Mouse because if you look very careful, these cells always have 2 ears. We have no clue what it means. We never saw them before. But the computer separates those out. It's bizarre.
Oh.
So you see things what the pathologist never saw before. But we have no idea maybe the pathologist looks at 7 or 8 cells and then he needs a coffee and he goes for the next patient. This platform takes a pool of 1 million cells and measures 100,000 cells. So your sensitivity is increasing. So of course the platform is going to detect things what you can't do by eye. So you need AI.
Absolutely. So going back to our question list here, this next question is for Kelly. I don't know, it might be a little bit specific, but perhaps it'll be valuable for other attendees as well. But one attendee has asked, we're interested in quantifying membrane expression of some of the targets on tumor cells. Do you have any thoughts or information about performing this analysis on DeepCell or other software?
So for this specific application, I would emphasize that our platform is label-free, so it's bright field imaging, so there are no antibodies used or fluorescence detection. So if the application did require labeling, surface markers with an antibody, it would probably not, be the best choice. However, if the presence of those targets would be expected, of course, to be a consequence of or lead to some specific change in phenotype or functionality of that tumor cell, which I would think would be interesting to explore with our platform and try to identify some of those mechanistic differences that are associated with those membrane changes.
Certainly. I'll let you decide who wants to take this next one, but an attendee has asked, would you always prefer UMAP via REM-I rather than CS data? And the context for this question, considering the fact that urinary cell carcinoma, apart from other tumor types of tumor, specifically microtumor niche for multiplexing spatial imaging and their overlap, UMAP data happens to be, what if it's completely different due to heterogeneity? I know that's sort of a many-parted question, but anything you could sort of add about, if you would always prefer UMAP via REM-I?
Kelly, you first or me?
Why don't you go first?
Okay.
We
basically have been looking
2 parts of the question.
Yeah. Okay, so I can comment on some of the things we looked into. We are very new in this field, but what is for us very clear, we want to only pursue changes that are totally independent on whether you make a PCA or whether you make a UMAP or a TSNE type visualization. So the more robust the changes are, the more clinically relevant we expect them to be. So your statistical methods should be sound, but your data, you should not overinterpret your data. You will always see differences. So if the differences pop up with the different methods as similar, that gives you way more confidence in your design of your experiment. At this stage, we are just in the learning phase. So we first, our body has something like, I think, 256 different cell types. Now, we probably have for 15 cell types a lot of morphological information. So for the majority, we still even do not have a lot of reference data available. In future, I expect this to grow exponentially because you're going to run all kinds of body fluids in which cells are present, and you want to study what's going on in these body fluids where suddenly cells start popping up. You can study lung fluids, you can study whatever you want. Eye drops, tears, spinal cord fluid with cells in it. So for many, many disease areas, there are lots and lots of different applications. So the number of cells, the more cells you have available, the easier it is to train your model and to detect it. If you only have a few cells in your specimen, then it's harder to basically study it. So I already got questions from dentists who want to study the immune response around the inflammation which is occurring near their implants. Yes, of course, those are very hot areas, but you will have fewer cells, but the type of cells that you are going to see can still indicate with what medical problem you're dealing, whether you're dealing with an infection or a response on an implant. So definitely there are different things visible once you know what you're looking at. And by first looking at the easy low-hanging fruit with the transplant rejection and the subtypes, once you know that, you're going to see also in these smaller areas parallels, because the immune system with its inflammation will not work different between the different parts of the body. It's fighting host pathogen responses and also other external triggers. So superimposing this data in the long run is going to deliver lots and lots of new opportunities. But that's why this human foundation model is so key to basically capture and learn and get more smart. And that basically will pay off in the coming years when more people are going to use and train the platform for their specific questions. Kelly, your turn.
I think you said it all.
Oh
my goodness, you're too kind.
Amazing. Well, this is sort of, this next question is sort of related, and, you know, I like this question. I'm focused on sort of big picture. So what is the value of improving morphology detection accuracy by 1% in terms of, you know, clinical outcomes and economics?
And who do you want to address this question to? Caroline.
I think that's one for you, Peter.
Oh my goodness.
Yeah, why don't you start?
Okay. Well, the cost of a bone marrow transplant is typically 150,000 euro in Europe. Okay, so if you study simply AML, acute myeloid leukemia, and you have to do a bone marrow transplant, there is a subgroup of patients which basically you do not have to give the bone marrow transplant because based on the mutation or the translocation those patients have, you can treat them with a drug which probably costs 12 euro at the pharmacy. So if you can recognize those patients more quick, more sensitive, etc., you will not make a mistake. So the translocation you can detect easy. If there's a point mutation in that same gene, it's way more hard to detect it. By having this kind of platforms where you can separate the cells, but also immediately do the downstream analysis to map these mutations. Yeah, if you basically can prevent that you lose a patient because of a bone marrow transplant, you switch off the immune system. If the patient gets an infection, he's dead. So that's a risk. If you don't need to do the transplant and you know you can treat with 12 euro, you do the 12 euro. Otherwise, the transplant costs 150,000. But if you basically can optimize these kind of decisions, it's like losing an organ what is transplanted. The transplant procedure costs money. We see differences, for instance, between the transplant of the kidney and the transplant of the heart. We currently think that it has to do with the selection procedures that are being used for the criteria to transplant an organ. The criteria are way more stringent in the kidney than in the heart because there are less hearts available. So you just give a little bit more immunosuppressant and then the thing will not get rejected. But if you have a better fit, of course you get less rejection. But you do not have the luxury of having a better fit because there are not enough transplant hearts available. So it's always you have to compare apples with apples and pears with pears. So it has to be really a true comparison. But losing a transplant organ and having to take a new one, the whole procedure has to be done twice. And if you can detect with this platform, we're dealing with an infection, you throw some antibiotics or some antiviral treatment, you don't have to do you don't have to transplant again. So the transplant procedure in total is a lot of money. So if a drug, take a little bit, a cancer treatment with a biological, the drug will easily cost 100,000 to 150,000 euro per annum. Okay? So if we now look at immunotherapy, we can see the T cell, which is going to find the other cell, which is mutated. And then you see doublets, because these cells, they look for each other and then they start interacting. So if you do CAR T cell therapy, you can actually see your repaired cell going to the damaged cell. And so monitoring your immunotherapy is immediately showing therapeutic effect. So those procedures where you can prove that an expensive drug actually gives a therapy response, you can count your circulating tumor cells by just looking in the blood, how many circulating tumor cells do I have? And then over time, by collecting 3 times a snapshot of what's going on, you already can monitor your therapy response much more accurate than what you could do till now. Because no pathologist is going to look at 100,000 cells, and especially not 3 times a week. So the throughput and the sensitivity, that's going to give big win-wins in the future. So it's a very promising platform. And again, it's not so fancy with all the colored dyes, but that's not the aim of the platform. It's separating and quantitative, get a feel for what's going on. The more advanced level you do and in the follow-up in the downstream projects. So this is why I think this will become a very valuable platform, which will be used in front of many machines. The proteomics platform, the DNA sequencer, the RNA-seq procedures. So you're going to make it more sensitive and more tailored to what's going on. And complex somatic mutations are hard to identify and to find at the moment. We think that by using this technology, subtle morphological changes can be detected much more easy. So probably the differences in the molecular signatures that you obtain after separating these cells, those can point to complete novel underlying molecular mechanisms.
Absolutely. Another question, any other ideas on applications in the ICU setting?
Yeah, well, that's where you have, of course, a lot of infections. So in ICU, if you draw blood and you want to have a better criteria to release a patient from your ICU, well, if you release him because of your gut feeling or you count his cells in his circulating blood, which point towards an infection, I rather rely on my immune signature that can be measured in the blood than the gut feeling of the clinician, well, the infection will be gone now after a week of antibiotics. So releasing a patient based on a change in immune signature, for instance, there can be a criteria to basically say, okay, now I'm confident the patient can get off from ICU towards normal care, or goes from high care to low care.
Mm-hmm. Certainly. Question on the size of cells. How big of cells are able to be analyzed on the REM-I?
Kelly.
Yeah, so the system is compatible with cells ranging from about 6 up to 25 micron. We have done some larger cells like cardiomyocytes, for example, that tend to, take on kind of a smaller overall morphology than you're used to seeing when they're, in a culture and spread out. So that's basically the upper limit.
Mm-hmm. I think we sort of touched on this, but it might be worth just taking this question again. So, you know, do you feel this approach would be effective beyond kidney transplant for profiling other types of organ rejection? I know, again, you sort of touched on that, but would love to circle back.
So we are very eager with what we have seen in the kidney transplant arena now. So the first thing we are going to do is go for a totally different hospital where they also have these transplant rejections. And we want to have samples from them, and we want to see whether they also have Mickey Mouse cells in their pool of cells. Because then we independently can see that, okay, the procedure of isolating your PBMCs should be identical. So you don't want different procedures because then you don't know what you're quantitatively comparing. But if they use the same protocol, you expect that in different centers you see similar immune responses. So that's what we would like to first, from our test cohort, go to a validation cohort, see the same type of morphometric changes using the REM-I platform. And then basically you want to start sharing the protocols that we use to isolate these cells and to preserve them. Because, okay, we had frozen down in our biobank the samples from these PBMCs. You very elegantly have to defrost those cells and then image them through the machine. That's something totally different from when you isolate the PBMCs and they have never been frozen down. So if you have frozen them down, you have to very elegantly defrost them and bring them through the machine. And well, basically one other aspect, what I think is interesting to discuss here, a conventional pathology laboratory was basically embedding everything in paraffin. And basically they used a lot of formalin fixed tissue. Well, that's something totally different from fresh frozen tissue that comes from the operating theater. So the formalin fixation introduces changes on the morphology, but also DeepCell is interested in dissociating tissue in future. So this is where we have huge archives with tissue blocks which go back for more than 40 years. So very well documented cases. If you can take from these paraffin blocks material which you can dissociate and image, you can start to find whole new concepts. So we think there is so much potential for this platform. And of course, there will be implications if you have formalin fixed, but the formalin fixation has the same effect on all the samples that have been paraffin embedded, formalin fixed. So among the samples, the effect is the same. So if you see differences, those differences must also exist in the fresh frozen, but then you have to collect a whole series of fresh frozen samples. So it's not said that you cannot use this platform on formalin fixed dissociated tissue. It's very indirect, but it's definitely possible.
Mm-hmm. Absolutely. Let's see. I think we have time for maybe one or two questions. Let's see. This one should be relatively straightforward. How many cells can be loaded at a time, and what resolution are they being imaged at? You know, like, for example, 10 times, 20 times, 40 times, etc.
Yep.
Okay,
so
Kelly,
you can load
do you want to address that?
Yes, sure. You can load up to 3 million live cells and up to 10 million fixed cells, and those are being imaged at 63x magnification.
Excellent. All right, let's take one more question here. Question regarding, can you perform metabolic profiling using this platform?
Okay, you can do downstream analysis where you look at metabolic signatures. For now, the microscope images the cells. So you don't use dyes, and you also don't use biochemical markers. So you can use on the separated cells, you can start measuring with your downstream analysis, of course, the metabolome. So you can either throw them on a mass spec or on a GC, or so definitely you first separate and then you do your downstream analysis.
Absolutely.
So it's a good question, because also metabolomics is adding a lot of value, but it's multiomics nowadays, as we said in the beginning of the talk. So it really positions it pretty central, this platform.
Certainly, all the omics. All right, that's all the time we have for today. Thank you so much. Yeah, we'd love to thank Dr. Peter von der Spek and Dr. Kelly Miller, and our sponsor, DeepCell. And if we didn't have time to get to your question, we will do our best to follow up with our experts and get you an answer. And if you missed any part of the webinar or you want to listen to it again, you'll be able to watch a replay available on our site. And just a quick reminder to, if you don't mind, completing the survey. All right, so that's it. Thank you so much again for joining us for this genome webinar, and I hope you have a great day.